You're listening to Just Medicine, an equity in healthcare podcast created by medical students in British Columbia.
Hello everyone, welcome back to the Just Medicine podcast. My name is Annie and I'm a medical student at UBC. You may have heard part or all of my 3-part series that I recorded recently on reproductive health. If not, you can check that out. But today we're here with an entirely new episode and a different topic, which is genetic database inequities. So I think this is both a very important and a very interesting topic, and we'll get into all of the details throughout the episode. But just to start, here's a little preview of what we'll be addressing and talking about today. So at the heart of the issue is the fact that you cannot just use the same reference data for everybody when doing a genetic test. Different populations of people have different background variants in their genes, so we can't just apply a blanket genetic database to everybody. The inequities of the situation become even further enhanced because some populations are very poorly represented in these big genetic databases, which obviously has big implications when it comes to genetic testing, diagnosis, and treatment for these poorly represented populations. We'll dive into all of this and more throughout the next hour, but just before we do, I'd like to introduce today's guest speakers. So we actually have 2 amazing guest speakers on today, both of which are involved in a really great initiative called the Silent Genomes Project, and we'll talk more about that project in a bit. So first, Dr. Anna Lehman is a medical director of the Adult Metabolic Diseases Clinic at Vancouver General Hospital and an associate professor of medical genetics at UBC, where she also trained. Her research program has focused on identifying genetic causes for rare diseases and on improving the diagnostic process for people with genetic disorders. And her clinical work focuses on improving care for patients with lysosomal storage disorders and other inherited metabolic diseases. We also have Dr. Wyeth Wasserman on the podcast, who is a computational biologist with a research focus on the human genome. His research lab, based at BC Children's Hospital since 2002, develops and applies computational methods, software, and databases to gain insight in the functional roles of specific DNA sequences in the human genome, with an emphasis on those that control when and in which tissues a gene is turned on. With the development of low-cost DNA sequencing, his lab has taken an active role in the discovery of genetic variation in patient genomes that cause rare genetic diseases. He also helped launch the Silent Genomes Project, which I mentioned earlier, to confront and overcome inequity in reference data for Indigenous children who need a genetic diagnosis. All right, so I've got Dr. Wasserman and Dr. Lehman on the podcast now. Thank you both so much for taking the time to be here today. Before we jump into the bulk of the episode, I'd love to hear more about each of you and what you do for work, what your interests are, and what has brought you to what you're doing today. So if you each just want to give a little bit of an intro, that would be great.
Sure. I'm Dr. Anna Lehman. I am a clinical geneticist, so I am a medical doctor who did additional training in the area of genetic diseases, and I subspecialize in treating individuals who have genetic conditions that affect their body's metabolism. I work at Vancouver General Hospital. I am also a professor in the medical school at the University of British Columbia, and I research genetic causes for rare diseases.
Amazing. That sounds really interesting. What got you interested in working in genetics in the first place?
I like the specialty itself for several practical reasons, including the opportunity to do a lot of research and incorporate evolving technologies, which have changed a lot during my career so far and will continue to do so. I thought it was an area that would have a lot of emerging treatment developments, especially gene therapy. So for me, I am a science fiction fan in my free time, and this was living science fiction that would help people in really practical ways.
Yeah. If you had to put in a plug, I know there's some future doctors listening to this. If you had to put in a plug for working in genetics, what would your number one thing be?
If the idea of never seeing the same thing twice appeals to you, then genetics would be a great field.
Okay, awesome. Thanks. And Dr. Wasserman?
I'm Dr. Wyeth Wasserman. I'm also a professor in the Department of Medical Genetics at the University of British Columbia with Dr. Lehman, but I am a PhD scientist and I am in a field called computational biology. So I spend my time using computers and computer methods to apply to the study of genetics. And my lab is situated at BC Children's Hospital in the BC Children's Hospital Research Institute. And I have the pleasure of trying to develop the computational methods to process the data that comes from these new technologies that Dr. Lehman referenced. And in that world, I get to see how the changes in computing and DNA analysis are moving along extraordinarily rapidly and move from new technology to new technology in my work. I did my undergraduate studies in mathematics and cell biology. And so there is a place for people who are on the data science side at the intersection of medical research.
Yeah, for sure. Something that we're going to chat about is that, well, genetics and genetic testing is advancing really rapidly. Not everyone has equal access to these things. So what drew your attention to that?
Essentially, we had a revolution in our understanding of human genetics about 15 years ago when a new technology was invented that allowed entire human genomes of individuals to be sequenced very rapidly. So this could be scaled up to hundreds of thousands of individuals pretty quickly. And we learned a few things pretty quickly. Unfortunately, we learned that many genetic variants that in the past we thought were causing disease and would lead to diagnoses for people were actually normal, benign variants occurring in the population. And we only were able to learn that after essentially reading the genome sequence of many, many people. And we learned around the same time that unfortunately for many of our patients, we did not have representative information about what was normal for their population. Of course, we're all related as humans. We all have a common ancestor at some point, maybe 200,000 years ago. And some of us are more related to each other than others. And so essentially we have subpopulations of people who are more related than others. And in the area of rare genetic diseases, when we're looking for a variant that is different and very rare, and in fact, maybe the patient would be the only one to have that variant if it is an ultra rare condition, it's really important that we know that we be able to say that with confidence. And so for each patient's own subpopulation, we have to have the representative data. And it became apparent that this was an inequity early on, as soon as these large data sets became available, and we were getting more variants of uncertain significance, which is a medical term that is given whenever we can't interpret whether or not a variant is a problem for a patient, is a diagnosis, or can be ignored. So at that point, it was really hard to know how to fix this, because it was such a big problem. But that's what drew me as a doctor to the issue, were real people in my clinic having ongoing problems with their diagnostic process because of this.
Mm-hmm. Would you like to add anything to that, Dr. Wasserman?
I'll just add that in the biomedical research community, one of the big opportunities is to sit together with people from different backgrounds and talk through things. And so for me, it came sitting in a medical genetics rounds where we had a genetic counselor and clinician describing a case and observing that this child was not able to achieve a diagnosis. And as we went further, discovering that there were variants of uncertain significance, too many of them. And as it went on, discovering that this child was Indigenous. And that was 2015, beginning of 2016, when I was sitting there. And at that point, you realize that you have a problem that's worthy to work on. And so I'm sure we'll touch on it later on. You know, it became obvious there, but then it's a long journey from that recognition to how you can actually address the problem.
Yeah, absolutely. And I think that genetic testing can be one of those things that's seen so clinically and so practically, but we have to remember that there are people behind those tests, and having or not having a diagnosis has so many implications. And we'll get into that as well later. But I'd like to take a bit of a step back just to start us off. Could you sort of explain, what is genetic testing? How do you go about doing that? And how do we use genetic testing clinically?
Sure. So first of all, an estimated 3% of us have a genetic condition. Many of us have a relatively common genetic condition like familial high cholesterol or hereditary breast and ovarian cancer. Some of us, though, have rare genetic conditions, and there are thousands of these diseases. And that's why altogether, even though they're individually rare, it's a lot of people who have and function and live their lives with a rare genetic condition. So genetic testing is a really important part of medicine. And in order to find out exactly what is going on with someone with mysterious symptoms, we often need to sequence their genome. A genome is just the set of all of their genes. We're able to do this more and more, but it's been a bit of a battle to get access generally because of cost. It's not a cheap test, and also the limited number of clinical geneticists in the provinces across Canada. So a person who seems to be having unusual health issues, often from birth, but sometimes not showing any signs until later in life, will be referred from a family doctor to a specialist, maybe a few specialists, before finally seeing a geneticist who will offer them the option of looking at their genes. And that's a question for consideration, because if you're going to look at all the genes, you might find other things that were not why you were doing the test in the first place. So that's a dialogue between a patient and a genetic counselor or doctor to decide to go ahead and do the test to see if there's a cause. We have millions of genetic variances or differences in our code. Essentially DNA is a letter code, and most differences in the letters have no physiological consequence, but sometimes they have very big ones. So then there's a process of interpretation and classification to figure out, well, which of these variants, again, are normal, benign, just background differences versus potentially causing this person's rare disease. So we have algorithms, we have what we call bioinformatic tools that use virtually AI to understand what the consequence of a change in DNA would be on the protein molecule, which is what the DNA is the code and recipe for. And then at the end of the day, we have a pile of evidence, and the evidence may be weak or the evidence may be strong as to whether or not a particular change in the DNA is causing the problem, is the diagnosis or not. And that's where all the pieces of the puzzle have to be examined together, and a decision is made. A decision is made with high certainty or low certainty, and the patient is involved in that discussion to understand if, do they have a diagnosis or not. And then from there, of course, the reason that we want to do that in the first place is to understand how that information can improve quality of life, healthcare outcomes for the individual. There may be a specific targeted treatment. There may be important pieces of knowledge about other health risks that could be prevented. There could be information about the chances for other people in the family to be affected. So for family planning, that could be useful. So there's lots of reasons that a person would decide to go ahead and have a genetic test.
Yeah, absolutely. Thank you. That was a really great overview. And like you mentioned, genetic testing is expensive. Could you give a bit more detail regarding that? In BC, are there any genetic tests that are covered, or how much would it cost to have your genome sequenced in BC?
That's a great question. So the first patient who had what we call genome-wide, which is this new technology, which would sequence all of the genes. This was around 2010, patient in Wisconsin, and it was $100,000. And the family fundraised for it, and a diagnosis was made that led to a life-saving treatment intervention for that child. So this case, of course, was making headlines around the world, and there was very quick translation into clinical trials for patients. And that's how patients in BC initially got a lot of access, was through clinical studies, what we call translational research, where you begin applying a new test or new treatment. And then we began getting some approvals to access the test by sending samples out of the country. where commercial labs were setting up in the US for the most part, and then a lab set up in Finland that we've been using. And it's only been quite recently that some provinces have been integrating it into our own healthcare system, so patriating that technology. Ontario has successfully done this, and other provinces like British Columbia are in a pilot process at the moment, and we will see where that goes. But this has been a long, slow process, but progress is getting made. At the moment, there are a few other tests that are done that are genetic tests. They're more targeted in the province. We have our pilot for the genome-wide, and then other patients can access the testing on a case-by-case basis through sending samples out of the country. That's where we are right now, and it will continue to improve. And in terms of equitable access, across the board in healthcare, we have issues related to things like rural location versus urban location, whether a person has a family doctor or doesn't have a family doctor. So all of these factors that we know impact access to specialty care also play a role for accessing genetic testing. So we continue have to meet all of our system-wide issues.
Of course, yeah. So it sounds like a lot more complex than just going for a test. There are so many factors involved. And I was wondering if you could speak to that whole process and all the uncertainty that a patient might undergo, having health issues, not knowing what's going on, and seeking a diagnosis, and what impact that might have, either being able to have a diagnosis and a management plan, or just being left with uncertainty.
What you say has a term in the medical literature. It's called the diagnostic odyssey, and it is studied, it is measured, it is a primary endpoint outcome that's people try to shorten and try in different ways. So different models of care have been proposed, and we call one popular idea right now is the concept of what we call mainstreaming. So getting other specialists, perhaps even family doctors, comfortable enough with genetic testing and making it fast enough and efficient enough that patients don't have to bounce from specialist to specialist for so long. I think of a genome-wide test, we use these terms, there's subtypes like exome sequencing, whole genome sequencing, as like what we call the echocardiogram, the ultrasound of the heart. In the past, it was such a long process for a patient to understand what was going on with their heart. Doctors would listen to the heart sounds, debate, take some x-rays, do some dyes, injections, and take more x-ray. You just do the MRI, you just do the echocardiogram, you just look and you just find out right away, not years later. And that's where we need to get eventually. And we are making progress.
Yeah, for sure. It's exciting that so much progress is being made, but at the same time, there are all of those extra reasons and population dependent reasons why certain people do or don't have access to genetic tests in the first place. So something that's really important in genetic testing is having these databases to compare genetic variants to. And I think this might be a question for you, Dr. Wasserman. Could you tell us a bit more about genetic databases and how they're created and why it's important to have so much diversity?
Yeah, it's a big question. I'll try to tell you. I always like to ground things in a little history. And so, genetic database has been around for a very long time in different forms, and it relates to the technologies that we have at the time. And so you start with the ability to detect certain things, and you build a database about that information, and then you can detect more things, and you build a different database that can capture all those pieces. So in the modern age of whole genome sequencing, you have a major database that's emerged called NOMAD, which is maintained by the Broad Institute in the United States. And the way NOMAD works is that they take in data that's been generated in research studies. So in a variety of research studies that have been developed, they will have sequenced the genomes of individuals, and then those genomes are made available in the NOMAD process, and they reprocess them in exactly the same way. So everything's analyzed in exactly the same way so you're confident in what you're looking at. And they count up how many times do they see each letter in the genome at each position. So the genome is 3 billion letters long. And so at position, chromosome number 1 at position number 1, you will have a count and so on. And that's incredibly valuable because now you've gone from not knowing nothing. That individual case in Wisconsin, there was no reference data, so you had no idea what was normal. And Nomad has grown. It's on version 4 right now, and it has grown to hundreds of thousands of genomes being available in it over a few short years. So I'm an incredible fan of Nomad's efforts, and they have tried as much as possible to capture more diversity in the efforts as they've gone along. But because of the nature of the data that they can get access, which tends to be data coming out of research studies aren't done equitably around the world, not done equitably with different populations. And so you get a bias towards what data resources are available to you. And so if you look at version 4 of Nomad right now, you will find that there's a breakdown of populations, and you'll see that there's a very heavy emphasis on European ancestry, fair bit on African ancestry, and that reflects that we find much greater genetic variation within people of African backgrounds, and that reflects our shared evolutionary history. So when you think about where humanity starting in Africa, that means you have the greatest variation present in Africa. But as populations move around the world and establish themselves, they will be a little bit of a selected pool. And so they'll have some pieces that connect back to that sort of historic times of Africa. But as you establish those new populations, they will develop their own characteristics in their population. So you have those subpopulation specific ones. And so around the world, as we've gone along, what people have realized is that Nomad's very good, it's very valuable, and it's incredibly helpful for interpreting genomes of anybody. So it's useful for everybody. But for each population, there's a set of genetic variations that you won't find in Nomad. And therefore, what you find is that different populations will start producing variation databases specific for their population. So for instance, in Denmark, they produced a Danish genetic variation database so they could better reflect the genetic variation that was found in Denmark. And if you look in the scientific literature, you'll find more and more and more of those types of studies, and each one is valuable. And it reflects that sort of gets a little cheaper. You can produce the data more readily, and you know that you can do better by your population if you have that genetic variation data that's suitable and specific. Nomad's still useful, but the more localized data makes it more equitably useful. And just to give you a sense, I would say you get about, and Dr. Lehman may want to correct me on this, you get about, I'd say 75 to 80% of the variation comes out pretty well from Nomad that's shared globally. But that still means that there's a big chunk of the variation that you're going to see that's more population specific.
Right. Okay, great. Thank you. So I think the main thing there that we're going to talk about today is that while there are so many different genomes, or so many genomes like number wise represented in those databases, you can't apply that to everybody. And like you were mentioning, when you do these subpopulation databases, that's more useful, but we don't have those for every population yet. And so we can't properly diagnose or, usefully genetically test every population because of that. And so that sort of brings us to the concept of the genomic divide. So when I say genomic divide, you two have obviously spent a lot more time in genetics than I have. What comes to mind?
I think it's exactly what Wyeth just described, in the lack of equal representation, the lack of diverse representation in these databases, and now that translates to less accurate test results and therefore worse healthcare, essentially. And that's unacceptable, right? That goes against everything we work toward in Canada as a country. So this is a priority. We can't, allow this to go unaddressed, or at least, acknowledging it as a first step. And then the whole process of trying to address it, though, becomes very complex. And I'm sure Wyeth will talk a lot about that. So for me, a genomic divide is a healthcare divide, and therefore we all have a responsibility, and especially if it's your own specialty, to do what you can to support change and improving care. And I'll also say, so the issue around, why there are fewer diagnoses for people who have less representation, absolutely it relates to not understanding, not being able to say what a normal background variant is, because then immediately, if you see a rare variant, well, you actually don't know it's rare. And therefore, and if that's one of the number one criterions for reaching a diagnosis and you can't use it, then you'll never get there. So that's essentially, how this algorithm works in variant classification. The other thing is that we see from studies in the US that there is a problem in the databases that track pathogenic variants. So subpopulations will often have their own pathogenic variants. So pathogenic means disease causing. And when there's the same disease causing variant in a population, we call it founder variant. So in other words, an ancestor first had it and it's just gotten passed down to everyone after that ancestor. And that gets big enough, but the people are staying in the same area, then it's a founder population effect. And we know for sure that a variant does cause disease if it's not present in people who are unaffected, and then it's recurrently observed in people who are showing signs of the disease. So you kind of need both things. And what they found for hereditary breast and ovarian cancer in the US is that clear diagnoses, so clear test reports, were way more likely to come back for people who reported their ethnic group as white than those who reported their ethnic group as Black. And the reasons, again, lack of representations in just these general pop databases, but also lack of access to the test itself. So when way more people of one ethnic group are having testing, then there's more recurrent observations of disease association. So these issues are complicated, and it starts, like as Wyeth was explaining, in who participates in research studies. And it also relates to who's having their testing done. And in the US, obviously, there's a big economic of who has healthcare coverage. And in Canada, we have our own issues with healthcare access. And that can be for many reasons. We have large immigrant refugee populations who might not have that literacy in terms of language, or knowing all the social procedures on how to access in a very difficult healthcare environment right now their care. And we have a lot of trust issues because of systemic racism and dark history. So if we have to begin then with learning and educating ourselves on history, and listening to what patients say about their experiences in the healthcare system. And again, if we're going to really address this, we've got to work on everything. So for me, the genomics divide, in my mind, says a very big thing from many different aspects of access, equity.
Our Silent Genomes Project lead is Dr. Laura Arbour, who's an amazing clinical geneticist who's been working with Indigenous peoples for 30 years on genetics. And so we look to Dr. Arbour for a lot of guidance. And one of the things that she's used as an illustration of the genomic divide is a metaphor of a bridge that she crosses when she's going to visit certain populations. So she works remotely in communities, and she will drive across this bridge. And so she has this wonderful picture to say, okay, you're driving across this bridge, you're going from these places of more urban settings of access and privilege, if you will, and you're going into communities that have less access. And so it's very literal, right? You can actually perceive this idea that you're going to places where people won't have equitable access to the specialist expertise and care. Now, one thing that wasn't mentioned, but I just think is incredibly important to take a side note on right now, is to say rare genetic diseases don't affect any one population more than any other population. So, your odds of getting a rare genetic disease are pretty much identical for no matter what subpopulation you come from. So that's we don't expect there to be any more or less genetic diseases overall for any population. We expect to achieve successful diagnosis at different rates, but we don't expect the actual raw numbers to be dramatically different.
And I'll add to that, just because it's so complex, just to add a layer, is that some populations will have certain diagnoses that happen more in their population than other populations. And this is true for all the populations around the world. And so we definitely want to know for each population, what are the conditions there? For example, newborn screening. Should we be including certain tests for certain populations? We're trying to move so hard to what we call precision medicine, which is really tailoring care to an individual in every way, which is efficient and improves outcomes. So we know, for example, the Ashkenazi Jewish population, we pretty much know all of the genetic conditions that occur at a higher rate in that population for the most part. And there are a lot of great targeted newborn screening programs and carrier programs and whatnot for that. And so we need to make sure that we're doing that for everyone equitably as well. But as exactly as Dr. Wasserman says, rare genetic conditions occur around the world, and our move to precision medicine, though, means that we do need to dive in and really tailor to which conditions for which populations. Things are all tied together, and as scientists and even as doctors, we always try to isolate little pieces and begin to think we understand, and then if you tug on one string, it pulls on something else. And so that's why, again, we've got to come at this holistically, and consider these everything, the environmental issues that come into play, the family history, the sociological issues that are at play. So you can never just address one thing. And even in this project, which we felt might have some tractability to it, we still there are many aspects to it that we have to involve.
Yeah, of course. And there's so much going on and there's so much to talk about. It can be overwhelming to know where to start. But you've both been working on the Silent Genomes Project, which we've briefly mentioned, but I'd love to get more into that now. So regarding the Silent Genomes Project, could you tell me a bit about what the project is, why it was created? And I know you have some very specific goals or things you're working on, so I'd love to hear about that.
Maybe I'll start and give a couple of things. So the Silent Genome Project, I mentioned Dr. Arbour. The other co-lead of the Silent Genome Project is Dr. Nadine Caron, and Dr. Caron is another inspirational leader. She's a cancer surgeon up in northern British Columbia, and she's suggested that Silent Genomes is ultimately about choice. And it's about giving choice to Indigenous peoples to decide whether or not they would like to participate and how it should be done, and not about what we think, but what needs to be done from the perspective of Indigenous peoples. When we launched Silent Genomes, we were thinking through it in 2016 and 2017 to put together the ideas, we emerged with 4 parts. So we recognized that ultimately what we wanted to be able to do was to give a choice to Indigenous peoples to say, do they want to see a reference genetic database created to help in the diagnosis of rare diseases? And to pursue that, the project was broken down into 4 activities, we call them. And the first one permeates everything, which is governance. And ultimately, how do you achieve Indigenous governance? What are the choices being made? How do you inform the work? And how do you align what you're doing with the best practices, if you will, in terms of Indigenous research? The second activity, which Dr. Lehman leads, is the applied clinical sequencing of genomes for Indigenous patients. And the idea being that even though we don't have a reference database, we should try to do our very best to diagnose we should try our very best to diagnose those patients that have rare diseases that haven't had an opportunity to have genetic whole genome sequencing done. And that has very profound and important things that Dr. Lehman will refer to, I think, later on. The third activity is the one I lead, which is the actual technical creation of the reference database. So how do we create the genetics resource that would be used in this way if there's a decision to move ahead? And the fourth component is around what I'll call the economics of it, but it's a very different take on economics because it's ultimately thinking about what are the values that we place on this. And so the values are not necessarily a cost value of how much are we saving the healthcare system in doing this work. It's much more, what is the perceived impact by the work that's taking place, and how do Indigenous peoples establish their values around genetic testing? So it's a very interesting approach to reframing the thinking around valuation. So it's not just about money, but it's about perception of how things are being done. That project launched in 2018. And so we have been going for 6 years now, and through pandemics and the like, with what I'll say was 5 years of consultation and discussion around the background library, establishing an Indigenous governance committee that met 35 times to go through and learn about the process and think about things and try to come to a conclusion about how this could be pursued. And early last year, early 2023, there was a determination that the governance was okay, that it was in a reasonable state, and that we should move ahead. And so we entered into a technical phase in the summer of 2023, and we have now coming towards the end of that technical phase where that reference library is poised to be completed and released. And we can talk a little bit about what that means later on. But I'm just going to check in with Dr. Lehman to see if I did a reasonable job of describing the overall scope of the project.
Did a much better job than I would have done. So Wyeth is one of the co-leads with Dr. Carone and Dr. Arthur. And I co-lead activity 2, which is focused on creating more opportunities for translational genomic diagnosis. So for families where whatever testing is available to them in the healthcare system has failed to explain what's going on with their child, we could apply the latest technologies, the newest, what we call bioinformatic tools, to see if we could find the cause. And we were able to do that in about 25% of the families who did participate in activity 2. And that's actually that's pretty good, given that during the course of the project, I'm happy to say their access improved in Canada. So many of them were coming to us already having had some pretty good tests through the healthcare system. Nonetheless, we were still able to solve a couple of mysteries for them and move them along in that diagnostic odyssey into just more management of the condition. I'll add too, another part of Silent Genomes is what we call capacity building. So at the moment, there aren't any Indigenous medical geneticists in Canada, and really what we want to have, is more and more leadership in our healthcare system, coming from individuals who are Indigenous, who are part of communities. And in my group, we have had 2 Indigenous graduate students, and one of them is now in medical school, and he says he's aiming to be a medical geneticist, and will hopefully carry on the work of Silent Genomes, which I think will need to go on for a while. Because at the moment, again, I think my role as non-Indigenous researcher in this area, maybe not ideal, and I would love to hand it off to Avery when he's finished, if he's listening to this. And we also are working on understanding how the what we call this library, the Indigenous Background Variant Library that Wyeth was talking about in activity 3, how that improves from a very quantitative standpoint the diagnostic process, the variant interpretation for our activity 2 families. So we can now go back shortly. We will be going back very shortly to reanalyze the genomes, and hopefully what we're going to see, I'm 100% sure what we're going to see is an incredible improvement in precision.
Yeah, I'm just going to add on the capacity building side, which is that, we're incredibly excited about the generations that are coming through the system right now. So there's some amazing young people that are emerging there. And one of the things we noticed in the field was that we didn't know of any Indigenous genetic counselors. I think there actually was one, but we weren't aware of them out there. And we know that there's now a few in training in genetic counseling programs. And so we can see the change happening in real time. You can see these amazing, bright, talented young people that are coming through the system. And so just thrilled that we can support their development and their work.
For sure. Yeah. It sounds like there's been a lot of years of working towards the exciting things that are happening now. And so that's really great to hear. I'd love to talk more about how you went about. So you said that now the Indigenous background variant library is complete. Did I understand that correct? Like you've gotten the information that you wanted to, or?
So I'm a computational biologist who specializes in information. I would always love to have more information because more information is always more useful along the way. But when you're going from nothing to something, we say it's an infinite improvement.
Absolutely.
So we are going from not having a resource at all to having a resource that's going to constituted right now in the initial effort from 4 communities across Canada that have chosen to participate and provide samples and have their DNA engaged. So we're excited that we have some diversity across the geographic space of Canada. And those 4 populations have provided a total of 600 samples. So we have 600 individuals, and each of us have a copy of DNA from mom and dad. So we have 2 copies of everything. So we have 1,200 copies of the genome that is going to be available in that database. We have gone through the technical steps. So we've sequenced that data. We've looked at the quality of that, and we were thrilled that we got really high quality data, which you don't always get. So sometimes things go well on that front. And we've done what's called the processing, applying the bioinformatics methods to that data to get it ready to count up how many times do we see A, C, G, and T at each of the positions. And now that has been completed, and we are now in the process of putting it into the database that the clinicians will be able to access to see whether the variants that they have. And we are at the point Dr. Lehman referenced very soon. We're at the point where I think Dr. Lehman's going to be signed into that system next week to be able to get in there and look at the data. Dr. Arbor's already into the system and starting to play. When I say play is not the right word, but starting to do some quality checks and make sure that it looks right according to her understanding. So it looks really good. We're very excited. And now after that, it becomes how soon can you get it in the hands of others? And getting to the data is something that there was a lot of discussion in the Indigenous Governance Committee around who should be able to access it and under what criteria. There's what I think was quite remarkable in that consideration was a recognition that international colonial lines don't mean anything when you're thinking about benefiting Indigenous people. So we have Indigenous nations that are split across the Canadian-US border. We have family relationships that go far beyond the Canadian border. And so I thought it was quite impressive that when the Indigenous Governance Committee was going through their considerations, that they concluded that the data should be available to benefit any Indigenous person who would be undergoing genetic diagnosis, and that those clinicians who are working with those patients, whether they be in the United States or Mexico or somewhere else, should be able to access the system, assuming that they thoughtfully understand the rules that are applied and what's allowed to be done and not done. And so there's a fair bit of education that each individual who's going to access the data has to go through to make sure they understand what's appropriate to do. And then as an extra aside, which I would just highlight for Dr. Lehman maybe to comment on, one of the incredible benefits of the activity 2, the clinical sequencing, was raising the awareness of Indigenous genetics and how to work with Indigenous patients more successfully across Canada. And so I don't know if Dr. Lehman wants to comment about the National Clinical Network.
Absolutely. So Activity 2 worked with the Activity 1 team, which was community engagement and Indigenous governance, to develop materials for genetics professionals, so including genetic counselors, medical geneticists, on cultural safety. So the process of hearing about a genetic test and deciding whether or not to undergo that process is complicated and time consuming, and it's really important that the person involved in this very personal discussion with the patient has that critically important background awareness, so that the person can be sensitive to and have an ear out, listen, be humble, and not making any assumptions about what's the right thing for their patient. And these materials, some of them are previously published, Truth and Reconciliation document, which I highly recommend to everyone to read. And some of them were created, a specific kind of genetic counseling guide. Also, a course that was created in BC called the Sanyas course was made available to everyone across the country. The cost was covered by the grant to the study, and we had close to 30 genetics professionals go through this entire curriculum. And so for us, this is a positive milestone from the project. And I think there was a lot greater understanding of this issue too through the project, which is important, because we still have more work to do. And also will say that Silent Genomes was partnered with an older important project in Canada called the Alliance for Healthy Hearts and Minds, which did quite a bit of biobanking with many communities, many people of different ethnic groups across Canada, and this kind of work takes decades, really, these kinds of changes in the healthcare system. So I think a lot of Silent Genomes was in trying to do things now and also investing for the future.
Yeah, of course. Thank you so much for everything you shared. It sounds like you're doing really amazing work. Sorry, were you going to add something there, Dr. Wasserman?
Just a tribute to the Alliance for Healthy Hearts and Minds in the top. So as Dr. Lehman referenced, this project has been going on for a long time. I think it started in 2012 for the Indigenous arm of the Alliance work. And they were instrumental in enabling our work to go ahead. So without their efforts, we would not have been able to advance the work. So it's a great project that people might find interesting as well.
Mm-hmm. For sure. Yeah. But yeah, it just seems like you're doing really amazing work and covering so many different angles regarding the project. And it must be so rewarding for you both working on something that takes a lot of years to make progress on, but will benefit populations for so many years to come as well. And that must also be exciting for you, Dr. Lehman, being able to go back and reanalyze the results of a genetic test with so much more information available. Yeah, that must be amazing.
Oh, I literally, for my own patients in my clinic, I have a list on my desk of patients, who have what we call the variants of uncertain significance, and as I said at the very start, one thing, as soon as Nomad was developed, we instantly realized we'd given a lot of misdiagnoses, wrong diagnoses to patients, and this is terrible, and so we've now been so cautious so I've been holding back on some diagnoses, until I can be reassured that I'm not going to give a wrong diagnosis. So I'm really excited for my patients when I can hopefully have some news, some updates for them very soon.
Of course, yeah, that might just be so satisfying and rewarding for both you and your patients. And just feels so good when you can deliver news with more certainty and not have these patients left in uncertainty anymore. So just to step back a little bit, in terms of Dr. Wasserman, when you were trying to broaden the information available and create the background library, how did you approach that? Did you have specific things that you did in order to keep cultural sensitivity in mind? Or how did you approach like asking someone whether or not they wanted to be part of it?
That's a very good question. And as a computational biologist, you discover that you can't do everything yourself. You are dependent upon a village of people to come together to make things happen. So I had this idea that we could create a background library that would be valuable from a clinical perspective. And so I started talking to some people, and they said, okay, you can't do this without Dr. Arbour. So I go and talk to Dr. Arbour, and Dr. Arbour also believed that the background library would be incredibly valuable. And she said, but we can't do this without Dr. Caron. And you start having those conversations as you chain this along, and you start going and having the conversation and seeing if you can engage the next person. Along the way, it was we have to be able to move the clinical work, and therefore we need Dr. Lehman. And so you start seeing that. Now, on the cultural safety side and how we do this, there is enormous bad history in research in genetics. And so when you're thinking about a project like this, you are doing it in the context of a history of abuses, of researchers coming in, saying they're going to do a research project and using the data for something else, doing research and not bringing the results back to community, doing research and using it to convey a community in a negative way when there was no reason to do so and it wasn't appropriate. And so you have all of these historical events that have happened. And when I say historical, we're talking about now, for 10 years ago, 15 years ago. This is not ancient history. This is real and aware in people's minds. And so when you have that type of situation, ultimately you have to engage extensively in conversation to say, okay, what are we trying to do? How can we listen to the people that we're talking to? So it's not us speaking at them, but it's really trying to find an opportunity to hear from people about what is valuable to them, and doing so in a way that is real. And so one of the things that you discover in this is that it takes a long time for people to get to know each other, to develop a level of trust that you can shatter that trust in a moment. So one of my concerns throughout this and every day is, how do I make sure that I live up to the trust that's been given to this, and how do I make sure that as a project we live up to that? Because it's so easy to see how things could go sideways and that you could lose track of what you're doing. In that process, it's about relationships. And so when we talk about how we were able to build off of the work of the past, it's Dr. Arbour spending 30 years working with Indigenous communities and advancing it. It's Dr. Caron establishing biobanking practices in Northern British Columbia and building relationships across Canada. It's having bodies that are willing to sit down and engage with us in conversation, whether it's the health committees of communities, or Assembly of First Nations health committees, or ITK. And each of these groups has been willing to sit down, have conversations, learn about what we're doing. And once there was sufficient understanding to say, okay, it's okay to move forward in what you're doing. And that takes enormous amounts of time. It means that you can't live on sort of a Western diet of Gantt charts and deadlines and deliverables. It means that you have to say it's going to take the time it's going to take, and we will proceed forward when we all agree to proceed forward and not before that. And so for me, a lot of learning around these types of perspectives, and how can you appreciate the perspectives of others, and trying to figure out how can you bridge between the way sort of Western's research functions and the way that each individual Indigenous community wants to approach the problem. And it's rewarding, but it's it's time consuming and doesn't move on a calendar that everybody can handle very easily. So we're very grateful, particularly to Genome Canada, which has been the main funding body for this, Genome British Columbia, because they've been willing to work with us and think about how can we pursue this project while reflecting that need to proceed only when it's appropriate to proceed.
For sure, yeah. And I think that's such a good reminder, not just within genetics, and not just with regards to this specific project, but in general, in terms of working in healthcare, though, meeting patients where they're at is so important, and you can't schedule in building trust. And obviously, I have very little experience with patients at this point in my training, but maybe Dr. Lehman, you want to comment on that and the importance of just being patient with patients in order to build a relationship where they're comfortable sharing things about their health and working with you, and trusting you.
Oh, that's the only way to practice medicine is to have, well, I guess we call it good rapport. I don't know if that term is still used, but
Still used, yeah.
yeah. So if you don't have that trust and that relationship, then you don't have a therapeutic relationship. You're not going to be able to help that person. And good intentions are sure, yeah, a necessary minimum, but that's nothing. You can't, good intentions don't get you very far. So listening and understanding and empathizing and going to where the patient is, in their mind and their thought process. And I also, I am a servant. I am in a service role. I have never thought anything else of, who I am and what I'm doing and why I'm doing it. I find out my agenda when I find out the patient's agenda. So, and this will get you pretty far, I think, in terms of having effective therapeutic relationships with your patients. And I am also very quick to respond when I have misstepped, and I'm very. For me, it's quick and easy to take a critical look at what I said. Was it, could it have been misinterpreted, or was it interpreted correctly and it was, an ignorant thing for me to say, and show that I can learn and grow with my patient, and get past, this and get back to solving the problems and moving them forward with their goals. So the added element is the colonialism and the systemic racism, in this project that we have to then layer into this and, really make sure who's driving the agenda and making these critical decisions about what we're doing. So I think Wyeth explained that whole process really well.
For sure. I think that there's a lot to be said for recognizing your own mistakes and just being honest with people, regardless of whether or not it's in medicine. And that's something we can all continue to keep practicing and getting better at. And that's amazing that it sounds like for you, it's very natural, very reflex at this point to be able to critically reflect in those ways. And I think that's so important, especially working in a profession within healthcare. So we are running a little bit low on time here. I would love to talk for much longer about this topic with you both, but I was wondering if you have any ideas regarding from an individual perspective, what we could be doing on our own to help move this in a positive direction. I know there's a lot of unlearning and a lot of then learning to be done, but I'm just wondering how you recommend we start as individuals.
Keep your eyes open to what inequities are happening around you in your field, in the hospital, and it doesn't have to stay that way. It doesn't have to be that way. There are going to be opportunities where you can make a difference in some way. And then at the same time, what we just talked about, every time you build trust with a patient and show them that you should demonstrate that you're worthy of their trust, then that's that's going to help their health outcomes, and that's going to lead to more progress with joint solutions and people willing to come to the table and work through the things that we have to.
Quite similar. I agree very strongly that there's lots of opportunities, whether it be in Indigenous equity or in other forms of equity. As you go through, you're going to see inequities all around you, and you have to be confident to speak up, call it out when you see it. And sometimes there's going to be something large where you have to decide, am I going to throw myself into this? Am I going to take it on? And you can't do them all, but there will be opportunities to get involved and make a difference. In terms of Indigenous equities, I do strongly encourage everyone who's working in the space to read some of these key documents that have been produced over time. The Truth and Reconciliation Report is incredibly valuable to read. It's not that long. You can work your way through it in about an hour to get a sense of what's there, and it will change your perceptions of what you think about. Likewise, there's some very good documents from the United Nations Declaration on the Rights of Indigenous Peoples, and those are also relatively straightforward to access and read. And I would say it helps you realize how things are and how we can do better. And just making those commitments to expose yourself to those topics is a step forward.
Yeah, thank you for that. Dr. Lehman, do you have any other initiatives or resources you'd like to mention?
No, I think we've covered the main things.
Okay. Well, thank you so much both for taking the time today to chat with me about this. I know the purpose was not to come to any answers, but more so just to talk about something that's important so I can learn more about it, and so that people listening can also learn more about it and become more aware of what's going on around them.
Thank you for having us.
Thanks for tuning into Just Medicine today. I really hope you enjoyed the episode, and be sure to keep your eyes and ears peeled as we've got a lot of other exciting episodes on the way. Until next time.